Yeah. Then we go to the next speaker, and that is Deborah Faria Fonseca. She's a PhD student. She comes from Brazil, far away, and she holds an MSc degree from the University of São Carlos in Brazil.
Thank you, Mr. Chairman, and good afternoon, everyone. This is part of my research for my thesis, and my advisor is the Professor Eugênio Foresti, and I will present to you part of the results. In the anaerobic digestion post-treatment, like the tertiary treatment, we can remove some compounds that are toxic or pollutants, like in this case, nutrients like nitrate and nitrite, and in the biogas, hydrogen sulfide. In the denitrification, that was the aim of this work, consists on the dissimilatory reduction of nitrate or nitrite to nitrogen with energy conservation. The main steps are in this figure, and the essential enzymes involved in this process are here in this figure. And the denitrification constitutes the global nitrogen cycle in combination with nitrogen fixation, nitrification, and ammonification. Specifically in this research, we studied the process called autotrophic denitrification, which uses the electrons of the oxidation forms like hydrogen sulfur to the process of reduction of nitrogen forms like nitrate and the nitrite. I'm nervous, sorry. And there's a great potential in this process by using reduction sulfur compounds, because it provides simultaneous removal of nitrogen and sulfur in a single phase system, and the conversion of these contaminants in environmentally acceptable forms. So the aims of this work, this research, was apply autotrophic denitrification process to anaerobic digestion post-treatment of anaerobic reactors treating sewage. Real here means not synthetic, and evaluate microbial communities established in denitrifying reactor with nitrate as electron acceptor and sulfide, hydrogen sulfide, as electron donor. This is the system of reactors, and 2 reactors in pilot scale. The first one is a baffled anaerobic anoxic upflow reactor for organic matter removal and denitrification. And the second one, it's an anaerobic nitrifying reactor and the samples are collected of the first reactor. Two conditions were studied here. The denitrifying reactor was feeding under two conditions. In the first, only of anaerobic effluent. In the second, with a mixture of anaerobic effluent and nitrifying effluent from the first reactor. We used the biomolecular techniques like PCR, DGGA, and the predominant phylotypes were monitoring using this based 16S or RNA genes fragments and sequence. We can see there was a selection in bacterial populations, and the richness has been reduced during operation in the denitrifying reactor in the condition C2, as we expect. This is the genus we are found. Bacterial sequences were clustered within Acidobacteria, Chloroflexi, Cupriavidus, and Haustonia. These genus are related to biodegradation and denitrifying activities and degradation of other compounds, pollutants compounds. And this, we can see images of microscopy of the C1 conditions and C2 conditions. The morphology includes rods, cocci, filaments, and sulfate-reducing bacteria-like. And this is an important result because after a period of incubation by this technique of estimation of bacterium population, we have a great selection by the media, which we can see bacilli were predominant. It's important because the nitrifying microorganisms commonly exhibit this morphology. And by this technique of quantification, we can see that results that is good results, because we have a very important quantification of these microorganisms, includes the autotrophic. So in the denitrifying reactor, about the denitrifying reactor inoculum, the effluent of nitrifying reactor presents sufficient amount of nitrate reducers, microorganisms, to inoculate the denitrifying reactor. And this effluent favored establishment and maintenance of denitrification in an oxy reactor. Richness reduced during the operation period may indicate enrichment of denitrifying populations. This technique provides an effective way for microbial identification of denitrifiers in environmental samples, such as sludge from a pilot scale reactor, like in this study. I would like to thank our agencies in Brazil, and to you, I would like to thank you for your attention. Thank you.
Thank you for the presentation.
Thank you, Mr.